Good afternoon, and thank you for attending Nona's webinar on direct CAR-based functional library screening platform to develop fully human heavy chain only CAR T cell therapies. Some of the highlights of today's webinar include optimized fully human heavy chain only antibodies are ideal for CAR T cell therapies. HCAb-VHs minimize immunogenicity risk and provide greater CAR T cell efficacy. The direct CAR-based library screening platform rapidly identifies HCAbs for CAR T cell therapy development. And without further ado, I'll introduce you to our speaker. Dr. Jason Noon is the principal scientist at Nona Biosciences and leads the research team at the U.S. site in Natick, Massachusetts, focusing on HCAb technology innovation, antibody discovery, and bioassay services. Before joining the company 4.5 years ago, Dr. Noon received his PhD from Iowa State University, studying innate immunity of host pathogen interactions. He then completed 2 postdoctoral fellowships at the University of Massachusetts Medical School and Boston University School of Medicine, focusing on mechanisms of both innate and adaptive immunity. Please welcome Dr. Jason Noon.
Okay, thank you so much, Larissa. So it's a great pleasure of mine to get to present to you today on the extension of our fully human heavy chain only antibody to CAR T cell therapy, and also get to share with you a very innovative and direct CAR-based functional library screening platform. So I'd like to begin this webinar by introducing the core of Nona Biosciences, and that is its Harbour mice technology. So the Harbour mice technology have been validated by numerous industry and academic partners. They've been applied in hundreds of discovery programs, many of which have entered the clinical stage. Harbour mice consist of both 2 heavy, 2 light chain format. This would be traditional antibodies, as well as a heavy chain only antibody format that is approximately half the size of traditional H2L2s. So the Harbour mice technology was invented by Frank Groesfeld. So he is also a professor at Erasmus University Medical Center in the Netherlands. And this here just notes some of our many highly reputable industry and academic partners. So among the core technology at Nona Biosciences is the transgenic HCAb Harbour mice. And these are, again, transgenic mice that can make fully human heavy chain only antibodies. And with the success of Frank Groesfeld's lab, these were the world's first fully human heavy chain only antibody transgenic mice, the first platform that was developed. And again, as mentioned, this has been clinically validated, this fully human HCAb platform with multiple HCAb-based antibody therapeutics in the clinical stage, whether a single antibody or bispecific antibodies. So the transgenic HCAb mice contain a highly optimized set of VH genes, and these VH genes are all derived from VH3s. And VH3s are widely known and published to provide the most soluble and stable HCAbs in the absence of the light chain. And the VH3s also have the most similarity to the camelid VHHs. So again, this is a very optimized set of VH genes in our HCAb mice. Also, our HCAb mice contain all of the diversity genes and also the joining genes. So the sequence diversity as well as the epitope diversity is very high in these mice. And we also can get from these HCAbs that are generated we can obviously get a fully human HCAb-VH domain. So this is like an antibody, it's just fully human. And now with this, Nona Biosciences has also launched the HCAb Plus. And what this is this is the extension of the HCAb to various therapeutic modalities, including bispecific and multi-specific antibodies, cell therapy, including CAR T cells and NK cells, and also ADC, as well as mRNA delivery. So the focus of today's webinar is going to be right here on the application of the HCAb, or the extension of the HCAb to cell therapy. So I want to start off by presenting to you an area that is becoming more clear, and a very important area in cell therapy, and that is the topic of immunogenicity of the CAR binding domain. So it's well known that the CAR binding domains are immunogenic, right? And they can elicit both B cell responses, so antibodies to the CAR, as well as T cell responses. And these B cell responses, these antibody responses to the CAR, these anti-idiotype antibodies, these can directly block CAR T cell function and also trigger numerous effector mechanisms in the patient when administered. And this can really diminish the CAR T cells and reduce CAR T cell efficacy as well as persistence. And these effector mechanisms are the well-known ADCC, ADCP, as well as CDC. Now, this brings us to an important topic here, where why has immunogenicity to the CAR binding domain not been addressed so much in the literature up until now? And it's quite clear that at least some of that has to do with the earlier CAR T cell therapies are targeting CD19 B cell malignancies. And it's interesting that in CD19 B cell malignancies, what is found to correlate with reduced CAR T cell persistence and disease relapse is actually T cell responses, not B cell responses. And that may very well be because the CAR T cells are in fact destroying the B cells and inhibiting the production of these anti-CAR antibodies. However, as we are moving out of CD19 B cell malignancies, in any way, even in multiple myeloma and BCMA malignancies, as well as obviously AML and solid tumors, we're now seeing that ADA does show a very striking correlation with reduced CAR T cell persistence and disease relapse. And that's in fact that the CAR T cells are no longer targeting the B cells. So there is a strong B cell response and strong anti-idiotype antibodies produced, limiting the CAR T cell efficacy and persistence. And this has been addressed very well and presented very well in this really nice publication in 2019 in PNAS. So in this study, there was a humanized camelid. It's a biepitopic anti-BCMA CAR T cell therapy. So there are two humanized camelid VHHs linked together by a G4S linker. And now this showed very striking efficacy early after treatment. And we see these stringent CRs, we see very nice before and after, the therapy was administered. However, you can see in the upper right that in the green, these are the stringent CRs. We see a lot of stringent CRs. However, then what you see in the circle is this represents those patients that still relapsed after the therapy. And what was found in these patients is that actually of all 6 out of 6 of these relapsed patients that showed diminished CAR T cells also showed ADA to the CAR binding domain. And you can see that this is at response and this is at relapse, right? And we can see here, we see that can range from around 11% ADA within that patient, all the way up to 50%, and then even over 90%, right? And so this overall ADA found in this study was 37.5%. So that's really high, and we're seeing that very strong correlation with CAR T cell, with diminished CAR T cells and disease relapse. And it's also important to mention none of the patients that showed good CAR T cell persistence showed any ADA whatsoever. So this was very strong support from this publication that ADA is going to be ADA to the CAR binding domain is a very important thing to consider for your CAR T cell therapies. And that brings us to this slide, where we can see very nicely from this Nature Reviews paper in 2021 that it's now well known that obviously CAR binders that have a fully mouse sequence are very immunogenic, and there's a lot of literature showing the impact on efficacy and showing the high ADA. And obviously you can humanize your mouse scFv or even camelid VHHs. However, even after humanization is not perfect, and you're still left with remnants of foreign sequence and still have that clear risk of immunogenicity, as exemplified in the PNAS paper that I shared. And now, what's better than at least a humanized scFv would be a fully human scFv, which again has a lower risk of immunogenicity, but still it is composed of two different domains. So you have twice as many CDR regions with the potential to have more immunogenicity, and also you have a linker as well. And all of these features can make that more immunogenic. So what Nona's fully human HCAb-VH will do is kind of the best of both worlds. It is fully human at its core, and it is also just a single domain. So this is the least risk for immunogenicity that we can get for an antibody-based binding domain. And Nona believes that this will be the kind of the best binder that you can have for your CAR T cell therapies. And to support the lower risk of the HCAb for immunogenicity, we can show some example data that we have for an HCAb-based CTLA-4 antagonistic antibody that used Nona's HCAb platform. This is in the clinic, had been dosed into 172 patients, and out of those 172 patients, only 7 of those patients, so only 4%, showed ADA. And there was no, even in these patients that had these, only 4% of patients that had ADA, there was no impact on the PK, on the safety, or the efficacy. So this is really supporting that reduced risk of the HCAb for immunogenicity, and including ADA. So now that we have an excellent foundation for using a fully human HCAb to adapt that and to apply that to CAR T cell therapy as well as other cell therapies, now I can share with you the data that we have collected along the way of doing this, of applying this HCAb to CAR T cell therapy. So what we have been working on at Nona Biosciences is, first we had an HCAb that had been identified by our Nona's more traditional antibody discovery methods, and we discovered this very good anti-BCMA HCAb, and we wanted to use this for kind of proof of concept in the beginning, and we cloned the VH from the HCAb into a proprietary CAR lentivector of Nona Biosciences. And you can see that here is our BCMA HCAb-VH in a CAR construct with the typical hinge or spacer, transmembrane domain, and intracellular signaling domains as well. And this is allowing the expression of the BCMA VH on the surface of the CAR T cells. So for the validation, the functional validation of this BCMA HCAb, we ran primary CAR T cell experiments by co-culturing these BCMA HCAb CAR T cells with a BCMA expressing multiple myeloma cell line. And what we observed was very striking cytotoxicity of the BCMA expressing multiple myeloma cells. We saw a very dramatic enhancement of CAR T cell activation only in response to the target. We're not seeing any activation in the absence of the target cells. And this is very much demonstrated by cytokine secretion as well, where we're seeing this is very much target dependent. We don't see any leakage, any tonic or non-specific activation or anything like that. It was very clean and very strong. So this was very encouraging for us to continue this. And what we have also looked at least in vitro, is we wanted to generate some data and explore the possibility of using our fully human HCAb-VHs for either bivalent, multivalent, or bispecific CAR T cells. And this was just an early piece of data that we got that was supporting this direction, where we had that same BCMA HCAb-VH that we made in a bivalent construct. So it's the same BCMA-VH, just there's two of them linked together by a standard linker. And we did see stronger activation of the CAR-expressing reporter cells in response to that BCMA-expressing multiple myeloma cell line. So this was very encouraging for us, and we're going to continue to go down this path of exploring or of utilizing our fully human HCAb-VHs for bispecific CAR T cells. So this brings us to the next topic for this presentation, and that is our establishment of a direct CAR-based library screening platform. So now that we know that our HCAb is going to reduce the immunogenicity of the CAR T cells and also show such great efficacy, we wanted to develop some rapid screening platform where we can quickly pick out the HCAbs that immediately from the data are going to provide the best CAR T cell function. So I can highlight the more traditional HCAb screening platforms that Nona has specialized in up until now. And firstly, I'll mention the Beacon single B cell screening platform. So this is such an excellent antibody screening platform. It is high throughput, and it's very fast. But a clear disadvantage, at least for CAR T cell therapy, is that it does not provide a direct CAR function readout, right? So it does have that limitation, and also it requires the use of specifically plasma cells. So you aren't able to include all of the B cells in the screening. Now, there's also phage display, and this does have advantages of being able to include all of the B cells. It is clearly a large library-based screening, but importantly, it is just binding-based screening, and it's not clear at all how well those binders will convert to CARs and show good CAR function as well. So it does have those limitations. And then thirdly, we have the HCAb direct expression system. This has those similar limitations of the first two screening methods. And importantly, it is not a direct CAR function readout, in addition to having a bit lower positivity rate. So we really wanted to develop something more tailored to CAR T cell therapy. And that leads us to this direct CAR-based functional library screening platform. So for this, at Nona, we have developed a proprietary CAR lentivector where we can amplify and clone in our HCAb-VHs into the CAR construct. Again, this has the typical hinge or spacer, a transmembrane domain, intracellular signaling domains, as well as an RFP for a transduction efficiency, and separated by a P2A peptide. And now, what we can do is with this platform is we can obviously do a simple mammalian display where we can stain the HCAb CARs with the fluorescently labeled target. But more importantly, I think for CAR T cell therapy is we can activate the CAR in a target-specific manner, and we can really customize this to any particular project need. And when the HCAb binds to the targets, this will trigger the activation of the CAR, triggering NFAT, and that is marked by GFP expression. And we can sort those GFP positive cells and recover the VHs and identify those leads for your CAR T cell therapy. So this has those obvious advantages of, it's very powerful. So we can use all the B cells. We can use all the memory B cells, all the plasma cells. So we maximize the coverage from the beginning. As mentioned, for the limitations of the other antibody discovery methods, this is a direct CAR function-based screening. So we can address whether those clones will show good and stable expression as CARs, and also good activation of the CAR as well before you even get that sequence. And this also provides multi-round functional screening to identify those leads. Okay, we can stimulate the reporter cell pool with parental cell lines to address non-specific binding, non-specific activation. We can address tonic signaling, and we can do multi-rounds of target-specific stimulation with recombinant cell lines, as well as various tumor cell lines as well. So it is a very powerful screening platform. Now, this slide gives you an overview of this direct CAR library screening platform. It begins by immunizing our HCAb Harbour mice. We then are able to recover the VHs and clone them into our proprietary CAR lentivectors and build the CAR lentivector library. We can then do the bulk packaging to generate the lentivirus library, and then transduce our JERCAT NFAT GFP reporter cells to generate the HCAb-based VH CAR expressing reporter cell pool. And then what we do is we can stimulate the reporter cell pool with target expressing cells, and we then sort the GFP positive cells in response to the target, and we can then recover the VHs and perform a next generation sequencing analysis and ranking. And in parallel, we do high throughput direct expression of these HCAb-VHs as a soluble HCAb, and we can confirm their target specific binding. And we do that for a number of clones, that then when we get those sequences, we can map those back to the NGS data to provide rigorous support for the numerous clusters that we generally see in the NGS data. And this really maximizes the amount of sequences as well as sequence diversity that we can get from this platform. So now I want to share with you our application of this screening platform to our own discovery that we've done as a proof of concept. So we've applied this in our very first project that we did. We applied this to ROR1 to see if we can get new ROR1 HCAbs that show good CAR T cell function. So for this, we immunized our HCAb Harbour mice. And we then isolated all the total B cells, including plasma cells, from high titer mice, and then we amplified the HCAb-VHs from the B cells, and then we cloned those into our proprietary CAR lentivector and generated our lentivector library. And then we did our bulk packaging to generate the lentivirus library. We transduced that into JERCAT NFAT GFP reporter cells. And without even stimulating the cells, just looking at what the GFP positivity is in the absence of target stimulation, we can already see some GFP positive cells in the absence of the target, and this would represent the HCAb-VHs that are showing non-specific and/or tonic signaling, right? And I just want to mention that this percentage is actually very low, and this was actually exciting to us, as it really is a testament to the suitability of our HCAb-VHs for CAR T cell therapy. And so obviously with this method, we can eliminate these clones immediately. And how we do that is then on the next slide, you can see we sort the GFP negative cells in the absence of target. the RFP positive, GFP negative cells. These would be obviously the CAR expressing but unactivated cells. And we always get over 90% post-sort purity. And then we stimulate those cells with the target antigen, in this case, ROR1. And now those GFP positive cells, in the presence of the target, we sort, and then we recover those VHs. And here we've sorted those GFP positive cells, recover those VHs by PCR. And what we did is we cloned those into our HCAb antibody expression vector, produced the soluble HCAbs, and then confirmed their binding, their target-specific binding. And from a number of clones that we identified, we moved forward with 3 of these ROR1 HCAbs for further validation in the CAR system. So, and what we did is we then individually cloned those 3 ROR1 HCAb-VHs. In this case, we're calling them VH15, 27, and 46. So we cloned them into our proprietary CAR-Lenti vector, and we also compare them to a negative control anti-lysozyme CAR, and then also a clinical ROR1 reference HCAb-V. And now what we can see on the bottom is RFP is marking our transduced cells, and on the y-axis we have the fluorescently labeled ROR1 staining for the CAR. And what we can see is that all 3 of our ROR1 HCAbs show excellent ROR1 CAR expression, similar to the ROR1 reference HCAb-V. And now what we can evaluate is the reporter cell, the CAR reporter cell function. And what we did here is we co-cultured these CAR reporter cells with different cell lines expressing different levels of ROR1. We had a ROR1 negative, ROR1 low, mid, and high tumor cell lines. And you can see that demonstrated here, whereas we have reporter cells only in blue, and then negative in red, followed by low, mid, and high in green, purple, and orange. And we can see on the left, this is our anti-lysozyme CAR. We see no activity at all. And then we have our clinical ROR1 reference HCAb-V showing nice activity with increasing levels of ROR1. And then we can see our 3 ROR1 HCAbs. They showed at least as good as CAR reporter cell activation as compared to that clinical ROR1 reference HCAb-V. And two of these ROR1 HCAb-VHs were actually better than that ROR1 reference HCAb-V. And this was really exciting for us then to take this to primary CAR T cells and compare their in vitro functions. So here on the left, we can now see again this is a cytotoxicity assay of the ROR1 high ROR1 high tumor cell line. And in the blue, we can see this is our anti-lysozyme CAR. And then in the red is our ROR1 reference HCAb-V, followed by our 3 HCAb, ROR1 HCAb-VHs, 15, 27, and 46. And now we can see our negative control in blue, and then we can see the cytotoxicity of the HCAb-V in red, and we can see that 2 of our ROR1 HCAb-VHs showed clearly superior cytotoxicity compared to the reference HCAb-V, especially this in green, this VH15. And this was further demonstrated on the right by the much greater cytokine secretion as well. So this was extremely exciting to us, and again, consistent with what we observed for the anti-BCMA HCAb, these ROR1 HCAbs are also showing a better efficacy compared to this clinical ROR1 reference HCAb-V. So moving on, then we also wanted to evaluate kind of the developability of these ROR1 HCAb-VHs as soluble HCAbs. And we saw here, you can see by both SDS Page and SEC HPLC, they looked excellent in terms of purity, so looking very good, very clean. And then we also looked at the hydrophobicity by HIC HPLC. And what we observe here is for these HIC retention times compared to Herceptin is that all three HCAb-VHs were very good, and these 2, especially the number 15 and 27, for which we did observe a better or superior efficacy. We saw very, very good hydrophobicity, showing just 16.7 and 16.9 minute retention time, which is really good. So developability was also very good for these ROR1 HCAbs. And then we also looked at the binding, the ROR1 specific binding for these HCAbs. And in the protein-based ELISA, all three showed high affinity, clearly sub-nanomolar affinities to ROR1. Then in our cell binding assay by FACS, we observed that while in purple, the 981, and then in orange, the 982, okay, these showed a bit higher affinity, showing sub-nanomolar EC50 as well as a low single nanomolar EC50. However, the 980 showed more of a medium affinity, showing an EC50 of around 10 nanomolar. And this was really interesting to us because this one in green is that ROR1 HCAb-VH that showed the far superior efficacy. And this is kind of consistent with the current understanding for CAR binders, is that really high affinity and low affinity aren't good. What we're really generally searching for is kind of a medium affinity, and most importantly, the higher avidity. So we're going to continue to explore what's really making this ROR1 HCAb kind of ideal for CAR T cell therapy, in addition to this kind of medium binding affinity. So that ends the topic of ROR1. Now, as kind of the final part of the presentation, is we wanted to extend this platform to a target that is a bit known to be a bit more challenging. This would be a multi-spanning transmembrane protein, and we also wanted to make this platform apply this in a more high throughput manner as well. So we all know that, or many of us know that Claudin 18.2 is a relatively difficult target. We need to, in order to get 18.2 specific binders, we need to target ECL1, extracellular loop 1, because this does have the different amino acids between Claudin 18.1 and 18.2, whereas ECL2 is identical. And in addition to getting 18.2 specific binders, we also want to see binding to cyno as well, and potentially mouse Claudin 18.2 as well. So this target is interesting and relatively challenging, and we wanted to see how our platform could do. So what we did is we immunized our HCAb Harbour mice, and at test bleed number 4, so this would be after several immunization boosts, we see in this cohort of 15 mice immunized with a HEC293 Claudin 18.2 cell line, we can see that all the mice show very nice positive titers, every single one. And what we did is we focused on group 1, a cohort of 5 mice, to build our CAR Lenny vector libraries. And that looks just like this, that workflow. So that's as I shared with you before, where we clone to the vector, make the Lenny vector library, do the bulk packaging, transduce the reporter cells, and then express the HCAbs, the HCAb-VHs on the surface of the CAR cells. But I want to draw your attention to what we did here. And in the first sorting, where we address nonspecific and tonic signaling, we co-cultured the reporter cell pool with a parental cell line to further address nonspecific activation of the CAR, and we sorted out the GFP negative cells, and then we co-cultured those GFP negative cells with the Claudin 18.2 expressing cell line, and then those activated GFP positive cells we sorted for further evaluation. And this was essentially an improvement in modification to the original platform. So this here shows the data that we got from the FACS, the sorting. So we can see before sorting in that first sort, where we co-culture with the parental cell line, we do see that GFP positivity in the absence of Claudin 18.2. So we sort the RFP positive GFP negative cells, and again get that high post-sort purity. And then we take those cells and we co-culture with Claudin 18.2 expressing cells where then we sort those GFP positive cells, again, get that high post-sort purity, and continue with further evaluating these clones. And now what we did here is we then recovered the VHs from those sorted GFP positive cells, and we cloned them into again, our HCAb expression vector to produce the soluble HCAbs. And for this, we did high throughput transfections of HEC-293 cells, high through production of the antibodies into the supernate, and then test the binding to the Claudin 18.2 specific binding. And what was really amazing, and we did quite a few 96-well plates to do this. And we were really impressed to see that our positivity rate at a minimum was 60%. And this was really excellent. This is a really high positivity rate compared to standard screening methods. And we even hit 80% in some plates. So, this was really exciting. And we're continuing to make improvements to this platform to even further enhance this positivity rate. But this is already very, very good and very efficient. So from those many positive Claudin 18.2 HCAbs that we got, we selected 24 here and show the data for binding to Claudin 18.2. We see all show, here you can see with CHO showing no signal, and then binding to Claudin 18.2 is really excellent. So this was great. And what we also saw, which was really interesting to us, is 4 of these HCAbs also showed binding to Claudin 18.1. Right, so this underscores that we are able to identify at least multiple epitopes at a relatively high rate from this screening platform. This was very exciting to us. So in addition to doing that direct expression in HEC-293 cells from our sorted GFP positive cells, after Claudin 18.2 stimulation, for which we know the positivity rate ranges from 60 to 80% already, we perform next generation sequencing analysis, and this would represent our data set immediately after sorting. But then we've generated a naive data set from unimmunized HCAb mice. And what we can do is any sequences that have high similarity or very high identity to naive sequences, we can filter out and get a much higher positivity rate, significantly higher positivity rate after there, and get the clean data set. And then we can do the ranking. And what we've seen is that our binders that we get from direct expression happen to fall after the ranking, happen to fall in those clusters that show very high abundance. Right, and this would provide rigorous support for all the clusters that we get in our NGS data set. So I want to show you what we did briefly for our NGS. Is, as I mentioned, we first co-culture the reporter cells after eliminating nonspecific and tonic signaling. We culture with a recombinant Claudin 18.2 cell line, and then we sort those GFP positive cells and generate our first data set, NGS data set, and then we continue to culture those cells and then stimulate them with Claudin 18.2 tumor cell line. And then we sort those GFP positive cells and generate a second data set. And then we can compare and identify those clones that are shared amongst the 2 data sets. And that shows the data here. And what we end up with is we get many sequences that are very abundant from that first data set. And then we get less sequences after then stimulating also with the tumor cell line. And we can do this interesting step called unification to enhance the sequence diversity. We also clean of naive, and then we narrow that down to a smaller set of sequences, and then we map them together. And then we ended up with 38 unique shared VH sequences with counts of greater than 100 within the NGS data set. So they have very strong support and relatively high abundance. And we can see that all of those sequences at least have several amino acid differences within the CDRs. So, and most of them much higher than that. So the sequence diversity is also very good. So we're really looking forward to the additional data that we're going to get on these 32 Claudin 18.2 candidate clones, especially in the CAR system. So that brings me to the end of the scientific discussion. So I want to leave you with some very important bullet points here. And that's first, that our fully human heavy chain only antibodies are highly optimized for excellent stability and solubility, and they are truly ideal for CAR T cell therapies. These HCAb-VHs, they minimize the immunogenicity risk, being fully human and being heavy chain only, and as I've demonstrated, also provide greater CAR T cell efficacy, especially compared to the standard the traditional HCAb-V binders. And finally, we have developed a rapid and robust direct CAR-based library screening platform. It is a direct functional-based screening for CAR T-cell therapy, and this will identify those HCAbs immediately meant for CAR T-cell therapy, having good and stable expression and also showing good CAR T-cell activation as well. So that brings me to the final slide, and that would be just to mention our innovative business model at Nona Biosciences. So I think this is truly amazing that we have no upfront fee, and we also have no royalty fee. So this is our open access for our technology, the Harbour mice, whether H2L2 or obviously the HCAb, especially suitable for a cell therapy. And for our discovery service, we offer a fee for service at any stage in discovery, and we also have a one-time milestone payment at the IND stage. And we will provide the full package of service from the target validation all the way to the clinical candidates. And we also have an FTE rate-based collaboration as well. And we do have our BD experts online as well. If you have further questions, I can refer them to our BD panelists. So that brings me to the end of the presentation. So I hope you enjoyed it, and now we can have the Q&A session. We can leave us on this slide here, and I can check and see how we're doing in our Q&A. We have plenty of time, about 10 minutes before the end of the webinar. Let me open up the Q&A here. So let's see what we have. Screening. Just looking through some of these questions. So I have a first question here. It is, what is the sequence and epitope diversity like for your HCAb and CAR screening platform? So I'm just going to mention briefly and reiterate that what we get from our NGS data is an extraordinarily high number of sequences. And we are able to customize the NGS data analysis in a way that we can narrow it down to the sequences of interest by setting thresholds for sequence identity. And by doing that, we can get a combination of numerous sequences, however many you may want, and also maximize the sequence diversity. And since the sequence diversity is so high, we're very likely to hit multiple epitopes. And I think that I showed some data on Claudin 18.2, where we have seen that we at least hit multiple epitopes. And I may have left out any important details, so I'm gonna also refer this to Musheng Bao, who is our head of biology, and he can add any further details on that. So please, Musheng, provide any additional comments that you may have.
For the HCAb mice, we have done quite a lot of projects. And from almost all the projects, we get plenty of very diversified sequences as well as epitopes. So definitely for sure, our HCAb platform can generate different sequences and also epitopes as well.
So Jason, can you hear me?
Yeah, hi, Joe. This is
Yeah.
our Vice President Joe Zhao. So he's has all the information.
No. I just want to add that the HCAb, even though the VH regions are limited by those 9 VH, but our D regions and the J regions are full complement. So we have a 24 D regions and 6 J region. They are full complement of DJ regions 1, 2. The HCAb mice has a fully functional somatic hypermutation. So vast majority of the diversity is really coming from the VDJ and the somatic hypermutation besides the diversity from the 9 VH regions.
Yeah, that's exactly right. Thank you, Joe. So we'll move on to another question. We have an additional question here. How does HCAb-VH stability relate to their expression in CARs and inherent tonic signaling? I can address this briefly by just reiterating as what Joe had mentioned a little bit there about the genes that are included in the HCAb mice. So I'll just reiterate that these are all VH3 derived VHs, right, which are widely known and published to have the greatest stability and solubility as HCAbs in the absence of the light chain. They also have the highest sequence identity and highest sequence similarity to the camelid VHHs. Okay, and what we are seeing now, as we've applied that to the CAR platform, is we are seeing a very strong correlation between transduction efficiency and staining for the CAR. So we are seeing that that is really indicating that the vast majority of the HCAb CARs are expressed on the cell surface, and that is very excellent in terms of expression. And then we're also seeing that the tonic signaling is relatively low, right? We as show we only generally see, out of a transduction efficiency of 10% or less, we're only seeing 0.1 to 0.2% tonic signaling. So that is very low, and again a testament to the suitability of our HCAb HCAb to CAR T cell therapy. So again, Musheng Bao always has better explanation than me, so I'm gonna refer this to Musheng as well if he has any additional comments on that.
Yeah, I don't have any additional comments regarding this, but based on our 2 campaigns, the tonic signaling or non-specific binders from our immunized HCAb mice are very low, below 5%. That's the only thing I want to add. Thank you.
Yep. Okay. Right, I'll move on to next question. This is, why do HCAb-VHs give greater CAR T cell function compared to HCAb-VHs? I mean, this is a great question, and this is something that we are so excited to explore. And so far, I shared some data where we at least see that our most superior HCAb-VHs don't have very high binding affinity, more of a medium affinity. So it's not that they just generally have a higher affinity. In addition to that, we have looked at expression of the CAR on the cell surface, and we actually observed that all the expression levels compared to that clinical ROR1 reference SCAV were all very similar. So it wasn't that the HCAb-VHs are just expressed more highly; they're in fact just very similar. So it's not high. It doesn't seem to be higher expression. And what we are continuing to consider is there's the obvious possibility that we may be targeting a unique epitope in that example that we shared for ROR1. That's one possibility, and the other, maybe even more exciting possibility is if we're seeing greater avidity, in these clones, and because as that's been mentioned throughout the cell therapy, the CAR T community, as being such an important feature of their CAR binders. So we don't know exactly why, but we're exploring that at the moment.
Yeah,
let's see.
Jason,
Yep, go ahead, Joe.
Jason mentioned two examples where we show VH-based CAR T behave better than the standard care of CAR T. But we also have a third example, also for solid tumor, where at least in vitro, Jason mentioned two examples of in vivo studies better than the standard care one. But we also have a third example, which is also a VH-based CAR T that behave much better than the standard care, the more well-known SFE-based CAR T in the in vitro setting. And we don't really know the reason, but those are the empirical experimental observations.
Yeah, thank you, Joe. So we'll move on to, I see I've got a couple of questions here. I'll address this this last kind of scientific related question, and then I think we can move on. I see another question kind of related to maybe the business, so I can refer that then to one of the BD panelists. So this last question here that I see is, can your direct CAR library screening platform be applied to other therapeutic modalities? So absolutely. It's not just limited to cell therapy. That we can do a mammalian display as well, and you know what we see from our platform is that, you know, it is that the majority of HCAbs that we identify from this platform so far that we've evaluated do have good developabilities in general, even as soluble HCAbs. So that is a testament of the suitability for just antibody therapeutics in general. And we can also mention about this multi-round stimulation and sorting that we can do with this platform. So we can really tailor the platform to any project needs, right? If we want to target a unique epitope, or eliminate any non-specific binding, you know, we can also do that, or binding to specific tumor cells. So, you know, that makes it widely applicable to all kinds of modalities. The final modality that I think would be really interesting to think about is for like T-cell engagers, because we can imagine that the clustering for CD3 and the TCR is going to have a lot of similarities between T-cell engagers as well as CAR T-cell therapy. So yes, I think it can be applied to other modalities. So we only have 1 minute left. So I see a question here. I saw one about the. I want to answer that. It's about the timeline. I don't see that question here anymore, but I do remember the question. It's, what's your timeline? Basically, timeline from start to finish. And actually, for that timeline, I think I'll refer that to Musheng first, and see if he needs to refer that to any BD panelist as well. So go ahead, Musheng.
Yeah, to the answer directly. Usually it will take 2 to 3 months. Majority of the time will be the immunization stage. If you have your antigen ready, so we can do the immunization immediately. Depends on the titer. Usually will take 2 to 3 months to get the binder. So if you want to do additional tests or to purify antibodies or do individual check, there will be a little bit longer time.
Yep. Okay. Thank you. Thank you, Musheng. So Larissa, we're now past 2 o'clock. So I'll leave it with you for the closing remarks.
Thank you very much, Jason. We would like to thank each and every one of you for attending this exciting webinar today. We'd like to let you know that we will provide answers to the unanswered questions here live. and we are very excited to talk to you later on. You can reach out to bd@nonabio.com and stay tuned for the next webinar. Thank you. Have a great day.
Thank you so much, everyone.